Hi students, good morning and welcome again. And I'm continuing the portion of excretion in life processes, and now we will talk about the excretion in human. So these nitrogenous wastes are mainly urea, uric acid, ammonia, etc., which are excreted in form of urine. So the unit of kidney is the main excretory organ, and the unit of kidney is the nephron, that is the specialized cell which is doing the function of excretion. So those cells are called nephrons, as you all know, and the other excretory organs with kidneys, associated with kidneys, are the ureters and the urinary bladder and the urethra. So when we talk about kidneys, these are full of these cells, specialized cells, which are neurons and of course capillaries. So if you want to learn about the structure of nephron, you have to simply remember that nephron starts with a cup-shaped structure, that is Bowman's capsule, and in the Bowman capsule there is a bunch of capillaries which is called glomerulus, and this Bowman capsule of the nephron will continue as tubule of the nephron. So nearer to the capsule, Bowman's capsule, there is a coiling of the tubule. This coiling is called proximal convoluted tubule, PCT. And then this tubule will run downwards and will form a loop, U-shaped loop, that is loop of Henle, and this is having an ascending limb and also a descending limb. So first of all it will descend to form the loop of Henle, and again will ascend to form the other coiling or bend of the tubule, that is DCT, distal convoluted tubule. Distal means at a distance away from the Bowman's capsule. So and this DCT, after DCT the tubule is connecting to the collecting tubule. Now the urine is formed here in this tubule, and then it is collected in the collected tubule. So how this urine is formed? First of all, this blood will enter through the arteriole into the Bowman capsule. So this is the afferent arteriole, and it will form a bunch of capillaries. Then the blood will be carried by another arteriole, which is efferent arteriole. So there is a difference between the diameter of the afferent arteriole and the efferent arteriole. So because of this difference in diameter, there will be huge pressure generated in the glomerulus, and because of this there will be ultrafiltration, which is the first step. And because of ultrafiltration, the liquid part of the blood will come out, in which urea, uric acid will be there, but along with a large quantity of water and other essential substances will also come out. So the liquid part has come out, and other substances have also come out with urea, uric acid. But now the blood has lost its composition, its viscosity. It has to be revived back. For that, the first coiling, the first bend of the tubule is PCT, where reabsorption of water from this first filtrate will take place, reabsorption into the blood, and along with water, the other essential substances will also be reabsorbed in the blood so that it maintains its composition and viscosity. Now this filtrate, after reabsorption in the PCT, reabsorption of water and other essential substances, it will run down through the descending limb of this nephron, and in the loop of Henle it will form a loop, as I said. There again reabsorption of water and some ions will take place, and this filtrate will again be carried to the DCT through the ascending limb, and in the DCT, if there some chemicals are still left in the blood, or some medicines are left in the blood, those chemicals will be, or minerals which are waste, will be absorbed in the final filtrate. So this final filtrate is the urine. Now this urine will be collected by the collecting tubule, and this tubule will carry this urine to the center of the kidney, that is the pelvis, and all these millions of nephrons are there in the kidneys. So the urine is being formed in each nephron, and then collectively this urine is carried through the ureters to the urinary bladder. And urinary bladder, when it is full, then there will be signal from the brain, and this bladder will be emptied. So when we get a signal to empty the bladder, so then the urine is released through the urethra, which is called the micturition. This process is called the micturition. So this is the formation of urine and how urine is excreted. There is a sphincter, of course, in the opening of the bladder, and also when the bladder is releasing urine through the urethra, so there are sphincters there in the opening of the bladder, so that when it is closed, when the bladder is full, the sphincters are closed. So with this you can understand how urine is formed and being released. So I have explained this in the classes, and I also have sent notes. So I think this video will help you in revising and understanding more clearly the process of excretion in human. Okay, so I end this video here, and will come up with some other important topics in my next video. So stay blessed. Thank you very much.